In this video, we will talk about what is transformation. Transformation is a part of genetic recombination in prokaryotes and the types of transformation that we can see. So now talking about what is transformation. Okay, so we will start with what is transformation. Transformation is defined as a transfer of free DNA released from a donor bacterium to the extracellular environment that is taken in by the other bacteria. So what happens here? We have two types of bacteria. We have a donor bacteria and a recipient bacteria. The donor bacteria will release its DNA, which can then be present in the extracellular environment and taken up by the recipient bacterium. So whatever characteristic is encoded in the piece of DNA that was present in the extracellular medium is now present in the recipient bacterium. So they have acquired new traits based on the type of DNA that they have taken up. So transformation is an example of horizontal gene transfer that happens in prokaryotes. So what has been taken up are pieces of DNA from the extracellular environment, which has come to the extracellular environment from the donor bacteria. These DNA pieces are known as naked DNA, which are dead or lysed, which come from dead or lysed cells. The genes that are present on these pieces of DNA can give the recipient bacterium beneficial characteristics such as antibiotic resistance or virulence. So we will start with the Frederick Griffith experiment to explain transformation. So Frederick Griffith is a scientist who was working on pneumonia. So pneumonia was a widespread disease present in the 1920s. So in 1928, Frederick Griffith was working on the causative agent of pneumonia, which is Streptococcus pneumoniae. So what he studied at that time is that Streptococcus pneumoniae have two types of strains. The first is a rough strain and the next is a smooth strain. The rough strain was non-virulent, whereas the smooth strain was virulent, which means the smooth strain caused the disease, that is the pneumonia, in the patients who are infected with the smooth strain. So why are they called smooth? Because they had a presence of a capsule which enabled them to cause a disease in the host. Now, he did a set of 4 key injections into the mice and then he observed what were the results. So first he injected live R strain. So R is a rough strain which does not cause a disease, and therefore the mice that were injected with the live R strain were present. That is, they survived. They were alive. The R strain did not die, did not cause the disease. Next, we have the live S strain. The S strain, when it was injected into the mice, the live S strain caused the disease pneumonia in the mice, and the mice had died. Next, we have the heat-killed S strain. So the S strain, which is a causative agent for pneumonia, was heat-killed and then injected into the mice. The mice did not suffer from any disease because the bacteria that causes the disease had been killed. Next, he conducted an experiment where he mixed the live R strain with the heat-killed S strain. So ideally, the S strain was killed and therefore the mice should not have acquired pneumonia. But what happened, what he saw was all the mice died. Now here he is confirming that there is something that is getting transformed from the R non-virulent strain to the S virulent strain. So the R strain is getting transformed into the S strain because it has taken up pieces of the genetic material of the heat-killed S strain that was present in the media. So once we kill a strain, it will get lysed, and the presence of the DNA pieces in the media can be taken up by the bacteria, which is now present alive, in our case, which is the R strain, and the bacteria will then transform into a virulent strain. So this is a summary of what he had done. So what he found as a result, he said that there is some type of transforming principle that is transferred from the dead S to the live R. He called this process as transformation. We have a pictorial representation of the same. Now, coming to the mechanism of transformation, transformation happens with the help of bacterial cells that have the ability to take up these pieces of DNA from the media. So the process or the bacteria which are capable of taking up these pieces of DNA are called as competent bacteria or competent cells. And the proteins that are going to help in this process are called as competence proteins. Bacteria will develop this ability of taking up pieces of DNA in later stage of their growth cycle. We're going to learn an example of Bacillus subtilis and how it undergoes transformation. So we have competence genes present in the bacteria's DNA that code for important proteins called as competence proteins. The competence proteins are named based on they have an example here. We have COM-EA. So EA represents the first gene in the fifth cluster. So we have clusters of genes that code for these proteins. We name each cluster in the alphabetical order. So COM-EA refers to the first gene in the fifth cluster. So now what happens? We have COM-EA and COM-G that help in binding the double-stranded DNA to the surface of the competent cells. So a competent cell is a cell that can produce competent proteins that can help them take up these pieces of DNA from the media. So whenever a cell has these competent proteins, that is when they undergo transformation. If not, we can chemically induce transformation to these cells, which we will look at in the further section. So we have the first protein, which is COM-EA and COM-G. These are two competent proteins that are going to bind to the double-stranded DNA present in the media, and they're going to help it attach to the surface of the cell. And then we have COM-FA DNA translocase. So FA DNA translocase is going to help the movement of the naked DNA into the cell. Next, we have the double-stranded DNA that is taken up inside is going to be degraded, and only one strand is going to be present. The single strand has been protected by a DNA binding protein called as REC-A. Now, REC-A and other proteins are now going to help the recombination of this piece of DNA into the host genome. So when we talk about this entire process, let's try to remember what is happening here. So let's say we have cell 1, which is my recipient in our case. I have cell 2 which has died, and I have the double-stranded DNA of cell 2 present in my media. So we have COM-EA and we have COM G. We have COM-EA and we have COM-G, which is going to help the double-stranded DNA attach to the surface of the bacteria. Once they have attached, we have translocase, COM-FA translocase, that is going to help in movement of the double-stranded DNA into the cell. So now what I have a cell which has a double-stranded DNA. The double-stranded DNA is now going to be broken down into single-stranded using the endonuclease enzyme. Now, once they are inside, they are protected with the help of REC-A. The REC-A protein is now going to help the single-stranded DNA integrate into the host gene. Okay, so they'll get integrated and there will be recombination that occurs. Now, the rate of transformation that is taken up is only up to 0.2 to 0.5% of the chromosome can be taken up. And whatever we have, the useful genes, there is very less chance that two important useful genes can be transformed together. So let's take an example of two genes A and B that is present on my DNA. There are very less chances that both A and B can enter into the new cell. Now, whenever they can enter, that means they are very closely present next to each other. And now we will be calling these as co-transforms, co-transformants, or the process will be called as co-transformation. So based on their nearness on the DNA, so if this is my DNA, based on their nearness on the DNA or their closeness on the DNA, they will be able to be transformed together during the uptake of the naked DNA. By understanding the mechanism by which they are taken up by the recipient cell, we can identify the location of these genes, and therefore transformation can be used in genetic mapping. Now, types of transformation, we have two types of transformation. We have We have natural transformation where the process is naturally occurring. The bacteria are capable of taking up the DNA on their own. We do not have to do any external help to them. Examples are Streptococcus pneumoniae, Bacillus subtilis, Haemophilus influenza, Helicobacter pylori, and Neisseria gonorrhoeae. Natural transformation, the cell has the following characteristics. They need competence factors, they take up the DNA actively, and the recombination is mediated by REC-A protein, which is a single stranded binding protein that is going to help in recombination. So here we can see the pieces of DNA that are present outside, naked DNA, are entering into the cell, and then they can be integrated and then undergo recombination. Now, coming to the mechanism, development of competence, then we have the binding of the DNA, entry of the DNA, integration, and then whatever DNA has now been integrated into the new cell will now express their particular characteristics, which is nothing but their protein production. They can give the recipient bacteria antibiotic resistance or important characteristics like virulence. Now, the second type of transformation is artificial transformation, where we are going to induce transformation through the in vitro conditions. These cells are not naturally competent, but we are inducing the process of competence to them. So we have chemical transformation, we have electroporation, and we have other methods which we can use. Now, we will start with chemical method. We are treating the cells with calcium chloride and other divalent cations. These are going to disintegrate this plasma membrane and then help them increase their permeability. So whatever DNA is present in the media can now be taken inside the cell because I have denatured or I have disintegrated the membrane stability. I can also use heat shock method where I will alternatively place the cells on high temperatures and freezing temperatures. So the constant change in the temperature is going to disintegrate the membrane proteins. Next is electroporation, where we are going to give an electric pulse to the cells. This electric pulse that we are giving to the cell will again denature or disintegrate the membrane integrity. This can work for bacteria, yeast, plant, and animal cells. It is called as electroporation because we are trying to create pores on the cell membrane using electric currents. Next is protoplast transformation. Protoplast transformation is a major method that is used in plant cells. We're going to disintegrate the cell wall of plant cells by using enzymes. We can use pectinase, we can use cellulase in order to disintegrate the cell wall, and then the naked DNA can directly be absorbed by the plasma membrane. This is used mostly for gram-positive bacteria as well as in our case, we're talking about prokaryotes, so gram-positive bacteria can be used. Protoplast transformation is also majorly used in the eukaryotic plant cells. We have a summary of the artificial methods. Now, why do we need to do this process of transformation or study transformation? So this is a method of horizontal gene transfer. So major characteristics like antibiotic resistance can be created with the help of horizontal gene transfer. They can adapt to environmental stress. They can create genetic variation. Genetic variation is the key for evolution, as well as we can study molecular cloning and recombinant DNA technology through this. So with this, we end this topic. Thank you.